Okay, I'd like to introduce Shihab Shamma to you. He's Professor of Electrical Engineering at the University of Maryland and Computational Science at the University of Paris. So he's got a joint appointment now, which seems like a really nice arrangement with parent labs in both places. Shihab, I think, is inspirational in the world of auditory neuroscience and computational modeling of auditory processing. And I first met him when I was doing my PhD at the first ever conference I went to. And so ever since then I've been inspired by your
so much younger.
So thanks, Shihab, for coming to visit us.
I changed the entire talk since yesterday. I was going to change the title, but I thought it would be too confusing because I didn't really realize the background of the people here. And rather than talk about really technical details and boring stuff like that, which is only interesting to Sue and me arguing, I thought I'll give you an overview of the kind of issues that we have to deal with when we study the question of how do we hear in a real environment. So real environment is you're in a cafe and there are lots of people around and you're talking and you have no problem focusing on the person in front of you and ignoring all the other sounds which could be even louder. That's real listening. And animals have to do this all the time. Birds have to do this. Frogs have to do this. It's essential in order to avoid prey, find mates, so on. So I'll try to convey to you a sense of how we study this and how we come up with hypotheses, how we test them. Everything I'm going to mention when I use the word neuron is really inspired by this animal, the ferret, which is now a fairly common animal in auditory neuroscience. It's also used a lot in vision. Much of what I talk about applies just as well for looking at the real world, where you have so many different objects around you and you have to identify one object away from everything else. And so that's what we call watching or looking or viewing in a cluttered environment. Okay, that's the ferret. Been a subject of human investigations for a long time. I found this sketch in a cave in the Pyrenees, which is incredible. It's 13,000 years old. And I think it's a ferret, at least. Okay. So when we do audition, that's a typical scene that I was trying to convey to you. You can easily have music in the background, somebody's talking, and when I'm talking to you now, my voice is bouncing all over these walls and roofs and ceilings and coming to your ear. So this person uses cognitive functions like attention to isolate one source and go after that. In doing so, they must extract a whole bunch of features of the sound. For example, the pitch of the voice, where it's located, the timbre or the quality of the sound, its loudness. And they have to take these and ignore the same features that come from a different source, like music, extract the target sound, the source, and bind them together in order to give them a unity, which we call then the source, or the auditory object. So you do this when you listen to music. We do this almost innately. We don't even think about it, except we really do, as we discussed yesterday, pay attention when we do this. But when you're listening to the sound, there's drums, there's guitar, there's a voice. All of these can be heard at the same time. And if I ask you to report, as a musician, an orchestral sound, you can easily select one line, for example, the second violins, and report and write down what they are playing. It's really remarkable. We have no engineering solutions that can do anything like this. Absolutely none. And everything that is out there is really kludgy, and it doesn't work well. And you'll see why in a minute. But there are two aspects about this process, just to move a little bit faster. One is about extracting these features that I described. And when you look at are you familiar with a spectrogram of sounds? Mm-hmm. Yeah? So this is the representation of sound frequencies as a function of time. This spectrogram, what you just heard, looks like nothing like the beauty of music and singing and all that. These are components that come from the different sounds, from the different voices. These big bursts come from the drums, all mixed on top of each other. That's what we have to try to do. Extract out all these different components and then put them together. The ones label them correctly. And another key aspect of this process is the sequential processing. That is to say, tracking a voice over time, or tracking the music over time, or even the beats of the drums. So these two aspects are intermingled. And the auditory system does this. Okay, so now this is a cartoon. How does the auditory system do this? It has to extract all these different features. Well, we study in animals where the sound comes into your ear. There's very early the cochlea of the ear, which is the place where you do the frequency analysis. Basically takes the sound and makes it distributed like a piano keyboard. And then, in parallel, the entire piano keyboard comes out. In parallel, all the channels associated with different frequencies go all the way up to your brain. And in the retina, for example, you have the retina, which is a very complex structure which has a lot of different types of cells and do a lot of processing. And then you have one more stage, which is called the thalamus, which would be right before the cortex, and then you go to the cortex. In the auditory system, we have many stages along the way. Many parallel pathways go to many processing centers, all up what they call the brain stem. And then they converge into this structure called the colliculus, which feeds into the thalamus, which is the gateway to the brain, where all sensory systems merge, or at least go through this gateway up to the auditory areas of the brain, the cortex. We think that all these different areas extract these different features, so perhaps these areas are involved in extracting location of sound, or at least the cues associated with the location of the sound, like the sound level differences and the delays between the two ears and so on. That's all measured in these areas. And pitch of sound, probably most likely extracted before you get to here. And then so most of the features are extracted by the time you get to the cortex, and then they arrive to the cortex, and the cortex does the heavy lifting of combining all of these and binding them to create the impression of a source, a sound source. That's the spectrogram, which emerges very early in the auditory system, right in the first stage, in the cochlear nucleus, called in this area. So this is a little bit simplified cartoon of what's happening. So there's the auditory spectrogram, which is, as I told you, extracted very early in the auditory system. Now, that plus location and pitch and I don't know what comes to the auditory cortex. That's the brain of a ferret. And in the ferret brain, this area is the primary auditory area where sound signals come into the cortex. So this is very important because that's where the representation of all the important features is. This area is very important. It was referred to in the previous talk. Is the prefrontal cortex. This area also responds to sound strongly, but only when you are thinking about the sound. Only when you're trying to interpret the sound, giving it meaning. Is it a warning sound? Is it a pleasant sound? And the prefrontal cortex projects back to the auditory cortex through many different pathways. Yeah?
Is there a general division between the ventral and the dorsal stream for auditory processes as well?
Yeah, there is a hypothesis like that. So I'll just tell you. There's a hypothesis like that. Whether it's true or not it's investigated, and some people find evidence for it, and it's not quite as accepted as it is in the visual system, but perhaps.
And one more question, you said all sensory information is relayed through the thalamus. Is it true for smell as well, or is it an exception? Because smell is not contralateralized, and so I was just thinking...
That goes to the olfactory bulb.
Exactly.
The bulb is cortex, actually.
Huh?
The bulb is cortex.
Yeah, but it doesn't go through the thalamus.
It doesn't, isn't it? So that's an exception, but I'm not sure.
Yeah,
about
I
it.
don't know.
Okay, so yes.
I should check.
No,
I use that
It goes directly from the nose
into the olfactory bulb, so it
Exactly, which is
bypasses the
interesting,
thalamus.
actually.
Yeah, well, the nose is funny kind of thing. But at least vision does, audition does, touch does.
It's important to ferrets, though.
Yeah. Ferrets have great hearing. Pretty good vision. I don't know about their touch, but because they work in the dark all the time, so they must use good smell. They do have very strong smell. I think the English know better about ferrets than anybody else in the world. You guys invented the ferrets. And actually, we cannot get ferrets from the United Kingdom. In Paris, we have to import ferrets from New York, which is ridiculous. And the people at Oxford who do research on ferrets, auditory research on ferrets, they get their ferrets from New York. I don't know. It's something to do with animal. Yeah, what? All you have to do is go to a farm and get them. And they laid out all the cables in London. The telephone cables, electric cables, and all that stuff. Ferrets used to be, that's why they were domesticated, to hunt rabbits in farms and also to lay out cables and tunnels. So you just tie a little wire and you send them down and they come out the other side. And they're amazing. They're easily trainable, and that's why we use them also. They're not like a cat. They're much more social than a cat. Okay. So the signals come to the cortex. Cortical cells respond to sound in many different ways. Some cells love to look at the spectrum of the sound and pick out the sounds that are very broad in spectrum, like the drum beat, for example, which has energy everywhere. And so if you were to think of a piano keyboard, if you were to hit all the keys simultaneously, some cells really are activated best by sounds like that, where others are very narrowly tuned. They respond only when you give them sounds that are like coming from a single key. And others are very fast. For example, they respond only to sounds that are very abrupt and die out fast, whereas others are very slow responders. They love to integrate over long sounds, like a violin sort of sound. as opposed to a piano. So you have all this rich variety of cells that basically parse out the spectrogram and lay it out in its all different richness. It's much like, I apologize, I can't turn off the sound, but that's my male thing. It's much like when you look with the eye, you can view things with sort of broad outlines by just squinting a little bit, or you can go close to a picture and look at all the details, and so on. So this is basically the view available to the auditory cortex of a spectrogram of a sound. Okay, and this is supposed to be a representation of all of these different dimensions that are available to you. So the dimension of speed would be here, the dimension of broadness of the sound or bandwidth is available, let's say, on this axis, and frequency is on that, and there are probably other dimensions like the pitch of the sound if it has a pitch. The location of the sound would be at another dimension. So one sound, one trivial, simple ah, can activate all these dimensions simultaneously, telling you where it's coming from, what pitch it is, what its bandwidth, shape of the spectrum is, how fast it is, and so on. All available simultaneously. So that's important because that's what we use in order to extract sound sources and track them. Now, I added a whole bunch of demos for those of you who are not auditorily familiar with all of this literature, just so that you appreciate that what you hear is not really what you think you're hearing. We do this in vision all the time. Visual illusions, auditory illusions are very common, but they're extremely important. They tell you really about how you're listening. So, just to orient you, for spectrograms, basically like that. So we call usually it tones, and these are the elements that we use in much of our studies. We play simple tones, and noise would be basically activating the entire spectrum of the sound. So this is what. And a simple vowel like a or e or oo is just a bunch of tones, usually harmonically related because they come out of a resonant box called the glottis here, or a violin versus a flute versus a piano would be also the same, but basically a bunch of tones. Some of them are larger in amplitude, so there's more energy in some tones than others. And if you change which ones have the most energy, you change the nature of the sound, and that's what we interpret to be a, e, and oo. And so this is synthesized tones like. Okay? Can you hear all this?
Yeah.
Okay. So this is really speech. Okay, speech is just a whole sequence of tones, sort of tone complexes that have energy profiles that change from one sound to the other. And you stack them one after the other, you get speech. Okay? Just to demystify speech, one in a different dimension, time dimension, if you think of a single talker, and this is one speech waveform coming out of the microphone of a single talker, you see these bursts of energy. Those are the different syllables that come out of your mouth as you speak. Okay? Those are kind of like a sequence of tones. That's also burst of energy, quiet, burst of energy, quiet. Okay? And when you have two talkers talking simultaneously, and usually, hopefully, saying different things, then you can paint one talker as green and the other talker as pink, and what you see is that because they're saying different things, they tend to not be overlapping too much. So people talk in each other's silences, even when they don't intend to. Speech is quite sparse. Sparse in the sense that it's quiet a lot of the time, here, 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 but then there's big bursts of sound that come. And when two talkers talk, and they're saying different things, then you get these looks into each other's voices, or clean looks, we call them. That is really just saying like the voice alternates between talker pink, green, pink, green, pink. And that's just like two tones alternating. Okay? So to simplify it. And finally, when you take one syllable, like ah or pa or gi, that syllable has a whole lot of features that burst in energy at the same time, representing the pitch of that syllable, like the pitch of E, the location, and the timbre. Is it E or A or U? These different vowels are just sounds with the same pitch, same location coming out of my mouth, but with different timbres. And that would be no different than having bursts of tones that have different, for example, pitches that would be harmonics with different fundamentals and different complexity, and the tones can come from different locations and so on. Okay? So speech is no more and no less than a sequence of complexes that have features that are associated with their pitches, and when you listen passively, you're not paying attention. You're asleep, and I'm recording in your brain or in your cochlear nucleus. Two talkers talk. It's like just having two sequences of tones. But if I ask you to attend to the pink speaker, to the pink voice, then what happens is, and this, we know this in humans, recordings in the brain of humans and the brain of ferrets, the pink signal becomes large compared to the green signal, which begins to be very weak or diminishes or disappears for that matter. And when you hear tones, as I will make you hear in a little bit, and I ask you to attend to the to the green, for example, well, what happens is exactly the same. Initially, you hear both, but then when you attend to the green comes into your foreground, and the other one goes to the background and goes away. You can record in the brain of humans and animals, and that's exactly what you see. Okay? So the cognitive function, one that of attending to a target speaker, basically translates to the target becoming salient and the others becoming soft and going into the background.
Yeah, in visual perception there is the invisible gorilla, Simon
Yeah,
Chapis, and
exactly.
for auditory perception there are invisible auditory
Completely,
gorillas as well.
all the time. Yeah.
Which is very
No, I mean, when you're listening to two voices and I ask you to report the telephone number that the speaker A is saying, and then afterwards you report back to me correctly the telephone number, I ask you what the other person said, you recall nothing. Absolutely nothing. So now I'm just going to give you a sequence of illustrations, demos of how this process happens. Okay? And some of it is innate, and some of it is attentionally driven, cognitively driven. So when you listen to a sequence of tones, and this is called the galloping sequence, this sequence of tones, those tones are close together a little bit in frequency, and they will sound like galloping. You will hear exactly what you see. And we call this a single sequence. It's a single horse, and this horse is going to stay a horse no matter how long you hear this. Okay? Nothing morphs, nothing changes, nothing happens. However, if you take these tones and make them far apart a little bit, further away, what you hear, it completely changes. Initially, you hear the galloping rhythm, but after just a little bit, the galloping rhythm vanishes, and instead you hear two uniform sequences in the first few seconds. Sue made a career out of proving this is wrong. So don't ask her about this afterwards, okay? But I think even she agrees that over a few seconds, you can tell for yourself, okay? That you hear instead, the galloping rhythm vanishes, you can't even get it back within a few seconds. And instead what you get is two sequences. One high that is going slow and one fast going low. That is, you get the red sequence and the black sequence. So tell me if I'm right or Steve. You still hear me? Who heard the two sequences emerge and then you hear the slow and the fast? And who's normal? Okay. I guess the majority did. 55 to 45. We're in the UK still. Okay. And so, this happens kind of really, it happens, at least in the demo that I gave you, spontaneously. You don't even have to think about it. This is really strongly driven. What's happening here is that these tones are far apart, they live in different channels, and they are not at all, they're alternating, they're uncorrelated, they occur at different times, just like those two speakers were. You remember? Yeah.
What is your explanation when people hear that as integrated, and they can switch to segregated and integrated even in a short period of time?
Well, the one before was integrated, and it never split apart, right? This one?
Well, it was really...
This one was a galloping horse, all the time.
I mean, I maybe if we listen to it long enough, do
Okay.
some people.
I mean, if you listen long enough to anything, you begin to hallucinate, and I have no explanation for hallucinations other than to, wave my hands and say, oh, there's adaptation in your brain or something. But this is really normal listening. This is within seconds, and that's long enough for any speaker to finish a sentence.
In mathematics, to convince somebody, it's a good idea to go to the limit. If on your next slide you increase difference in frequencies even more.
Oh, then it's definitely, then it's even harder. Then it's even harder. According to Sue, you can still integrate it, but of course, yeah. So actually, you could still integrate it. For example, it's well known. Even the boring classical people who think this is segregated and two separate, they will all tell you that if you lose attention for a second, if a poster falls, and suddenly you're distracted, and then you come back to hear this, it sounds galloping again for a second or so, and then splits apart. So whatever that is, losing attention and coming back to it, could happen spontaneously when you hear for a long time. And then you kind of lose the focus, whatever that means, and then you get it. I have much better demos coming up. By the way, yeah.
Is there a threshold for this?
Special?
This threshold.
Threshold, what do you mean by threshold?
When it starts happening?
Typically about 4 semitones or so, This is the number used.
It varies with time.
Okay. Details aside, at about 4 semitones, and depending on the rate and all that, you start getting clear splitting. So if it's 6 semitones or 12 semitones, splits readily. If it's too slow, it does not. So if it's 1 tone a second, for example, it does not. Okay? So you have to go at about 3 or 4. By the way, not coincidental, that is the rate of the entire brain. That is the rate of speech, that's the rate of visual tracking, this is the rate of anything in your brain that you do is about 5 to 10 hertz. This is the speed of consciousness, if you will. And that's where all these things happen. If it's too fast or too slow, you go outside of that range. And then different things apply. Okay? Yeah.
Just a question. If I hear kind of at the same time, integrated and just me, what do it fit in the...
You know, as I said, you could hear it integrated, you could hear it split depending on if you lost attention, if it's too fast or too slow. You know, different people are different somewhat in their inclinations, and this is psychology. I was going to say this is not science, but this is dangerous. It's too dangerous. Okay. Yeah. So, you know, the exact amount and number and all that would vary depending on the complex listener that was doing the listening. Yeah?
Is it related to the statistics of natural sounds at all? So what counts as a natural
Well,
sound?
it does in the sense that, natural sound?
Well, you know, I think a couple something, you know, it's got certain relationships with
Yeah, as I said, natural sounds that we perceive and enjoy and all that stuff all have those kinds of rates. And it's because that is the physiology of our brain. And so that is, for us, the acoustic world that we care about, which is also the rate that we produce in the motor system, which matches that, about the rate that we like to run, about the rate, it's all of it's all related, because it's the same brain that hears and produces.
Regarding the psychologist, we know that there are pathways that goes that way from the cortex to the cochlea.
None of this stuff really is related to anything that these pathways do. These pathways are rather slow. They probably do engage in long-term habituations, learning, but long. I mean, for example, even in the brainstem. The brainstem is only 2 steps away from the cortex. When you record there, and see, I distract your attention, and if I play that galloping rhythm, it will be gone. Okay, so even in the brainstem, which is massively receiving input from the cortex, 10 to 1. I'm not exaggerating. There's 10 to 1 projections from the cortex to the colliculus, then from the colliculus up. But when you record in the colliculus, you see that minuscule changes with attention. There might be some changes, but it's like you're so proud you're showing there is effective attention at the colliculus. It's so small. Whereas when you're in the cortex, just 2 synapses up, huge. And if you go further into the areas of the cortex, away from the main input region, which is called the primary area, you go to the secondary or tertiary areas, huge changes. When you go to the prefrontal cortex, it only responds when you're not only attending, but you're attending with a certain meaning to the sound and so on. So the rule is the further away you go from these early auditory stages, the bigger the effects of cognitive states are. So the cochlea, really small. Yeah, so don't invoke the cortex to explain anything at the level of the cochlea. Those 2 tones that I showed you, A, B, A, B, if they're connected, you cannot split them because you are really perceiving a sequence now. And just now I have to move a little faster because I want to get to the other demos. I called these tones A, B, A, B, and I gave you a demo. However, exactly the same thing happens if you replace this axis with anything. So it could be A, B tokens of vowels, for example, oo, you know, and you get exactly the same galloping rhythm and the whole thing. If there were different pitches of complexes, tone complexes, you get exactly the same thing. So this axis can be anything, for as long as the percepts are separated enough. So pitches are separated enough, timbres are different enough, locations are different enough. No matter what, you get the same story. Okay? And long before we were ever born, and our parents are born, and our grandparents are born, musicians knew this, and they used it to make beautiful music with the, especially Baroque music, when you have melodies playing on the high line and the low lines and playing. They were written exactly like these. They're basically pitches that varied rapidly and far enough. And you split them, and you contract one or the other and play different melodies in these. Okay? So, and I don't think Telemann probably played a long enough stretch to integrate in Okay. What I just want to emphasize now one other aspect of this problem, which has to do with, for lack of other words, but let me just use this word initially, the synchronicity of sound. A source, when it produces sound, all of its features arrive to your ear at the same time. So when I say, for example, ah, my voice produced sort of these pressure waves that get into your ear, and at the same time, on all the different pathways that have to do with the location of that burst of sound, the pitch, the frequencies that are composed, that compose the vowel ah, all of these are activated simultaneously. And so that's a very important cue that tells you this ah, with all of this multitude of features, all comes from the same source. Okay? So synchronicity over time is what I use the word coherence, is, or temporal coherence, is hugely crucial, important concept in segregation. So this illustrates that in some sense. It's sort of, I'll play you first sounds which is the black. So first you'll hear you're hearing the black sound, the top line. Now you'll hear the lower line with the red tones, identical frequencies and everything, but they're interspersed between the black lines. When you hear this, you have no way of knowing where the black tones are anymore. The periodicity of the black tones vanishes. Do you get what I'm saying?
Yeah.
Okay. How can you recover that? How can I make the black tones? Visually I can, by painting these red and these black. You can easily see that the black ones are nice and periodic, and those are randomly scattered in between. Auditorily, of course, you can't. But I can do that by, for example, adding a bunch of tones flanking the red tones. And now, it's trivial for you to hear out the black tones, the periodic black tones, and the red tones do not interfere anymore. The reason is because you can scream the red tones and the black tones apart because they have different timbres, different spectra. So let's hear that. First you'll hear the black tones, and now with the red tones interspersed, try to hear them. Okay? So it's real easy because you can filter out the different timbres and maintain. very much like a voice, except the black tones don't even need to be regular. It's just easier to explain here. But it's because they have different features, and these features then, and all these tones are bound together, and they are not coincident with these, so you can easily segregate. If I just take one of these tones and make it a little bit off, that is, take the flankers and move them, desynchronize them from this, then these flankers do not grab this tone anymore, and it will again screw up the percept of the black tones. Okay? Now try to hear the black tones in the complex. Okay? Now the flankers fail to grab the... And this is another really nice demo. These are all due to Bregman, who's done a lot of these demos. Basically in this demo, just very quickly I'll tell you, I play you this two tones, these two tones, they're nice and separate and alone, you have no problem hearing them. But now, if I put this, so you hear this going down, these two going up, but if I add these black tones with these black tones now screw up the regularity of these going up, and so you hear instead going up and down, and you cannot hear these very easily going up anymore, because what you hear is the entire complex. So how can I recover the red tones away from the black? I want to get rid of the black and segregate it out. One way I can do that with the flankers, like I showed you earlier. But another way to do that is to create a context or a stream, or to make them belong to a different voice in advance. And then you will group them aside, away from these, by simply presenting a bunch of black tones beforehand. That creates for you the stream or the source of black tones, and that's precisely what happens when you start following a voice. And now all these other distractors, or the other ones, segregate away. And you hear easily these separated from these, okay? So let me just play these quickly. And then we're done. So these are the pure two tones you can hear, down and up. So you lost this one. And now, in the final demo, you recovered immediately. Okay? So this illustrates the time dimension, the ability to group things along a sequence by continuity. And the only reason you can do that is because these tokens, these tones, these pitches, these whatever they are, they share the same properties. So if you speak and you change your pitch by an octave every quarter of a second, nobody can follow you. But the reason we can follow you is because you don't hop around at 4 times a second. You don't change your pitch at 4 times a second. All of these properties are constant, quote unquote. And that's why you can create a stream and you can segregate my voice and follow it. Yeah.
Just in the third example, do they have to be in the same?
No, they should be roughly in the same channel.
No, not
within
the
2 or 3.
frequency, but the pattern.
No, they don't have to be regular.
They can be random.
Yeah. As long as they're roughly of the order of few hertz. So that's important. In other words, if you make it too slow, you don't do it. It doesn't work. So if this was an hour earlier, obviously it doesn't work. But even if it's a second earlier, it doesn't work. Okay?
So basically, it's an example of Gestalt principles of continuity.
Yeah.
which are applied to
Yeah,
auditory
absolutely,
perception.
absolutely. And this is all happening very quickly, and that's going to be the second theme of what I'm going to try to demonstrate to you with the demos, is rapid plasticity, rapid adaptation in your brain. As you listen to anybody, you're immediately integrating the information over the last about second or so. And that influences what you hear at this instant. And those are connectivities that form extremely rapidly. When you engage in a task, in attention, it's not going to happen really. If you just walk into a room, or the famous room of the cocktail party, and you don't pay attention and you just drink cocktails, no segregation, no cocktail party problem. I mean, only cocktail party drink problem, I guess. But if you go into a room with a lot of people talking and you attend to one source, whether it's the music or a single speaker and so on, all of these adaptive effects happen immediately. Immediately, that is within a fraction of a second. I'll show you just some demos of that. I have some demos that are really amazing due to colleagues of mine from Paris, which illustrate that. How you can hear the same thing in completely different ways by even involuntarily. Okay. When do you guys want to have lunch?
Well, 10 minutes, I think. Yeah.
I'll just play the demos and then we'll quit. This is the spectrogram of speech. And for the longest time, people told you that the most important about them is the peaks, where these peaks are, because the peaks tell you, like I told you earlier, tell you that this is vowel A, this is vowel E, this is vowel U, and so on, where the peaks in the spectrum are. They even give them separate names, special names. They call them, for speech people, they call them formats, and so on and so forth. Well, it turns out that if you pull out these peaks, and you make speech like that, it's called sine wave speech. So at any cross section here, you have a lot of energy at the appropriate place for a vowel or for the sound that generated this. But now I threw away all the other weak stuff. All these other weak tones that were around, I threw them away. And I did also something else here that was not very nice. I made it more continuous. I tracked where those, oops. I basically drew a line here, and that's what I played you. I'm going to play you. And there was another line here, and another line down below. So it's very continuous. None of this change breaks, not too many breaks. So now when you hear this speech, it sounds like a bunch of whistles. You hear that? I don't think many people probably, if they haven't heard it before, could hear what is said. Just play it again. Okay. Yeah. However, one thing that might rescue it is that if you instead, actually, if you make breaks in this and just simply play like the piano once every half a second or second, slowly, and you play this, it sounds like actually a nice melody. See? So there's an example. So if you want quickly to compose a piece of music, that's one trick you could do. And it even has meanings, because the speech underlying it has meaning. However, if I play it faster, it begins to sound more and more like speech. Okay? When you begin to get more and more of the meaning. So this is, again, slowly. I can't even hear it here. Can you hear it? If you can hear, it's fine. I heard them enough. Sorry, did you want to turn it up? is, by the way, just as the, yeah, that's good. As you just said, this is the sentence, where were you a year ago? So here, so now you're all here.
Top down.
And this is more speechy, musical. And this is, again, musical because it's a slow rate. And now when you speed it up, it sounds more like speech. And anyway, there are a whole bunch of experiments actually to show how, but what happened, and the reason it becomes more speechy and more objectively tested, tested by people who tested this, more understandable, is because you create essentially common onsets, a lot of synchronous events, and those group these together, and that's where you begin to hear. When they were instead just a bunch of whistles, like that, there was no way for you to easily group this one with this one. And so you heard just like separate whistles. But as Sue Denham, who you may know, said, you can will and learn to integrate these and actually hear them in an intelligible way. So that's a demonstration of exactly what you say. That there are people who do PhDs on this, and they can perfectly understand this without any cues or anything.
What are the chirps on the next one? These individual...
Yes, these are...
well, I said not just the line, it's...
oh, because they were percussive, what do you call it?
Rock starts, yeah.
Yes, you get a splatter.
Okay.
Okay, let me just play you now demos about the other important aspect of hearing, which has to do with the adaptive aspect of how is adaptation, or I use the word adapt to mean change, evolve, rapid plasticity, or whatever you want to call it. When you listen, you're not just listening passively to sound. There is a lot of feedback coming down from the higher areas of the brain, from the prefrontal cortex and other areas, influencing the representation of sound and what you actually report. So let me show you a demo of that, which is really remarkable. So this is courtesy Presnitzer and Claire, who just got her PhD. She's doing a postdoc. So what you'll hear is sounds that go doot, 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 and then a little gap, which can be, by the way, as much as a minute. But you'll all leave the room if I put a minute. And then you'll hear two tones with a pitch change, which can be either up or down. So I want you to tell me the direction of the pitch change here of this last two compounds. Okay? So this is most people heard it up. You heard it down, I think. It's interesting. There's always the odd man out. All right. So I'll play it again one last night. Up, okay. And here I'll play this for you again, but just the last two. It's the identical sound, right? Up. But now I'm going to put a precursor, which is a little bit different. No. So these last two sounds, I'll play it again. It's down, right? And now I'll play you these two sounds here, just like you heard before. Down. But if I play it with the precursor. So this doot-doot or doot-doot completely changes. It's an identical sound. But it depends on what you heard just before.
When did you run this experiment actually?
This has been going on for years, like 3-4 years, and it's just being submitted. That's the French style, but it takes forever too. But there are countless experiments done on this now with all kinds of parameters and all that. It's a fantastic illusion. And but this illustrates that when you hear, I change your brain, not I, but the sound changes your brain. And it changes your brain is not the same after hearing something else. And therefore what you hear next is completely different. And how you interpret it. So that's exactly what we do when you have priming, when you have expectations, when you have all these things are effective because they change the way you perceive the world.
That sounds pretty psychological, actually. Is it science?
It's neuroscience. You can find all the neural correlates of that. So there is a scientific basis to psychology. Just to inform you about this. And here, this memory of that sound can be kept for, minutes. But if I distract you, if you distract yourself, if you wish it to be down really badly instead of up, then it can happen. You can change your memory. And you lose that effect. And you can find neuroscience associated with this because there's really good science to explain how you can do these rapid adaptations, how short-term memory is stored in the sequences and so on. And that's what you're doing. That's the last demo. That's what you're doing even when you're hearing speech. So this is synthetic speech. It's not quite as nice as the actual demo with real speech. But this is nice because it's completely controlled. You'll hear 2 syllables written here as arga and alga. Okay? Except, just like that demo I showed you, this last syllable is identical between the 2. But you hear it as da when it's preceded by r, and you hear it as ga when it's preceded by al. Again, there's some really simple adaptive reasons why the memory of what happened here persists and affects how you perceive this. And this is beautiful work of Laurie Holt at Carnegie Mellon that studied this. And you can hear demos of the real thing with it, but I'll just play you this. So this is the first one.
Arna, 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 Arna.
Okay, so you heard Arda, Arda. You didn't hear Arda? And this is the second.
Alna, 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 Alna. Arna, Arna, Arna.
Okay, so as I said, if you hear the real, this illusion actually originates with Ladefoged, who's one of the pioneers of speech research early in the 40s, 50s, 60s. Beautiful stuff. Anyway, I'll stop here just to illustrate these 2 mechanisms are always active, always in your head, and always doing the sound segregation for you. So, they're really simple, very simple mechanisms. There's nothing more to it than what I described with tones and all that stuff. But the only way we really can put it all together to explain how we can segregate and therefore build algorithms and things to mimic them is by doing psychology, psychoacoustic experiments with these simple phenomena to try to tease them out. And yeah, if you have any questions, I can deal with them during lunch or after lunch.
Thanks.